Hi, Kasia. Good morning. How are you today?
Hi, Chris. Good morning. Wonderful. Here it's a bit rainy actually in the Netherlands, so could be better. What about you?
Well, Switzerland is still sunny, I guess, for 3 days more. I wanted to ask you something. Today, I forgot what we wanted to talk about. You mentioned last week, but then I.
Are you talking about the Genomic Connections episode? It's
Exactly.
about it's bioinformatic.
Oh, bioinformatics, such difficult concepts. And how do you think you can explain this to our public?
Oh, well, you need champions to really go through this together.
Oh, talking about champions, do you think you have a champion in the eyeball community that can explain to us?
Well, sure. We have Rutger from Naturalis Biodiversity Center. I think he's pretty strong.
Huh. Our champion, I see him like the King Arthur of Erga. It's Diego de Panis from IZW in Berlin. I prefer to don't pronounce the entire name of the institutions because it's quite long and tedious to understand.
Wonderful. Well, I think we can get started, but I'm not sure whether the audio format will be enough for this very special episode.
All right, we went to writing blogs to making podcast, and now you're not happy with the podcast anymore. Let's try, we can try to make a video.
Wow, sounds wonderful. Let's get started.
Okay, let's get started. Name, team, and affiliation.
Diego de Panis, Team Erga, and the affiliation is Leibniz Institute of Wildlife Research, Field Forschung. Yeah.
Rutger Vos, Team Eyeball, and I work at Naturalis Biodiversity Center in Leiden, the Netherlands.
How would you briefly describe what you do?
I coordinate the sequencing and assembly committee of Erga.
I struggle with DNA barcoding data and try to get good stuff out of it.
Years in bioinformatics.
Not many. I am a biologist, so I'm just improvised in bioinformatics.
Bioinformatics exists since the dawn of time. In the Bible, Adam and Eve named the species, and that's the first step in bioinformatics. It's an ancient science, the first one.
Bioinformatics, more biology or more computer science?
I think it's a weird mixture of the two, but I will guess this more biology.
I'm going to have to agree. Yeah, at least what we do is more biology. There are fields in bioinformatics that are very algorithm oriented, but we're mostly just struggling with the mess that is biology, genetics, and the naming of species. It's all just very messy, and we're doing a lot of bookkeeping to keep that clean.
Coffee, mate, or tea while coding?
Mate, all the way.
Oh, definitely coffee always.
One thing that Eyeball does better than Erga?
Do we?
I think it's a better branding. They are more they're older, right? They're much older.
Eyeball has a huge community of people who collect specimens in the field. So I think there's many people behind it who just collect the specimen and want to have it barcoded. And Erga is probably just by necessity, because genome sequencing is a bigger endeavor, is more focused on fewer institutes. So as a community, Eyeball is bigger.
One thing Erga does better than Eyeball.
Everything. I think that since Erga is much smaller, or like the high quality reference genomes community, and the VP is much smaller, it can achieve things like more rapidly, like throughout the community and vertical integration of protocols and workflows, I think.
I think Erga has better integrated infrastructure. At least from where we are now, it looks to me, and I'm not sure if the perception is accurate, that there's more tooling already in place where Eyeball is developing more right now.
Are there any misconceptions people have about your project?
Misconceptions. The challenge is to make people understand what's this about, right? I don't know if many people understand what is important of having reference genomes or high quality genomes for the eukaryotic biodiversity.
I think a misconception with DNA barcoding is that it is more precise than it really is. Probably a lot of people think that it is a kind of a yes or no answer, and you can absolutely identify every species with a single barcode. And nature is more complicated than that.
What, you can't? You can't identify everything?
I'm afraid not, Diego. No, I'm sorry.
Favorite tool or software for data analysis?
I'll say I really like Minimap, and also I really like HiFiAsm. This is a tool for mapping reads to genomes and also for reference or for mapping reads or for generating genome assemblies. I think they are super solid software, pretty well written, super fast.
It's hard to answer. I can name some tools that I work with a lot. That probably not my favorites. We're trying to do more with Galaxy, so I'm learning more about how Galaxy works, and it is a very useful tool. I think I'm probably most excited to start up this tool called Mesquite, which is a kind of a graphical tool to explore sequence data and phylogenetic trees. But that's more also nostalgia, because I did my first postdoc with the Madison twins, who are the developers of Mesquite. And I also wrote some modules for it. So every time when I start it up, I see the credits, and then I get kind of nostalgic for my time in Canada, where I contributed to that.
Which is tougher, collecting specimens for DNA or assembling chromosomes on a computer?
Collecting a species for DNA, that's much difficult.
Yeah, collecting the species is difficult, especially when for the barcoding we try to do it obviously at scale, right? In the barcoding activities, we try to capture more species, but shallower. And so that is very decentralized and done by a lot of volunteers. And they have to then also, first of all, grab the right species, identify it correctly, and record in a consistent way what they've done. And when people input data, we make mistakes, right? So then obviously there's going to be mistakes in what's collected, and that's very difficult to correct afterwards.
Any friendly nickname to each other's team or project?
What?
I just call them the genome people.
And who will achieve the mission first? Millions of barcodes or genomes of all you species?
I would guess the barcode guys are. I mean, it's so easy to get a barcode right. It's just a tiny piece of DNA.
Exactly. We're already there. We have millions of barcodes. It's mission accomplished.
Describe yourself in one word.
Biologist.
Rutger?
Nerd.
And describe your counterpart in one word.
He's a bioinformatician, I would say. He's a bioinformatician, a proper bioinformatician.
I thought you were a proper bioinformatician. I'm just faking it.
No, I'm not.
So we are both, I think, by training biologists. And so when I say I'm a bioinformatician, I feel a little bit like a faker. Maybe you feel that way too, but that's not very flattering. Maybe say generalist. Like you can be both a biologist and have the technical expertise. So let's say generalist.
Bye. Okay.
The biggest myth people believe about bioinformatics?
I'll say that it's a thing that is super straightforward and effortless, that you can just do a few lines of code, and then you're going to get everything out of it.
Yeah, that's exactly right. It's the struggle is much more with just the messiness of the tools and the messiness in the data. and just struggling to stitch everything together. Also in such a way that we actually know what we did afterwards and why we did it and come to reasonable information. So it's much messier than people perceive it to be, I think.
If you could sequence any species next, which one and why?
Really good question. I think I will sequence something from the southern hemisphere, like a crown eagle.
I'm very excited about where ancient DNA sequencing is going. So the thing that just as kind of a passion popped up in my head is that, of course, we have the human genome and we have the Neanderthal genome, we have the Denisovan genome. I'm holding out whether it's going to be possible to sequence a Homo erectus. I'm not sure if we'll ever find good fossils for that, but that would be amazing.
Okay, and to finish, say goodbye and wish something to the other for the end of BG.
Bye-bye. Thank you very much. And I wish him a lot of luck, success, and
Thank you very much. Bye-bye. And yeah, I wish you many coherent assemblies.